once you've got a phylogeny that you feel really confident in, there's something neat that you can do. You can actually use the information that's present in the existing organisms, the present day organisms, and you can use that to go back in time and reconstruct what their ancestors were like. Here's the logic of how that goes. Let's take our vertebrates that you're very familiar with here. Oops. Let's take our vertebrates that you're really familiar with here, and let's look at mammary glands. All right. The chimpanzee and the mouse both have mammary glands. What does that imply about when that evolved? Well, it kind of suggests that this ancestor had mammary glands, but that this one didn't. So because if mammary glands evolved somewhere after this ancestor, you would expect only the mouse or only the chimpanzee to have them, right? So this was the latest that it could have evolved. If it had evolved somewhere back here, you'd expect all these species to have mammary glands too. So the most parsimonious reconstruction is that mammary glands evolved somewhere on this branch. Okay, you can go through and do that for any character that any of these species have. Okay? Another way of approaching it, you remember that one synapomorphy, like mammary glands, supports one clade. Here's the group of organisms that share the synapomorphy. This implies that you can stretch that box back past the most recent common ancestor for that clade. It implies that this ancestor here was the first to have that derived character state. And so the tick mark that denotes the evolution of that character state ought to go there. Generally, this is how we denote evolutionary changes on a phylogeny. You do a little tick mark across the line that says, hey, this is the branch where we think this trait evolved, and you jot the name of the derived character state down below that or above that. There are different ways of writing it, but they basically all come out to the same thing. You're labeling the point on the tree where you think a particular character state evolved. And this is something that you could be asked to do on, for example, an exam. Remember that when you're doing a homoplasy, you have to put in multiple state changes. So for example, with a pigeon, you need to show that warm-bloodedness evolved here and that it evolved there. Okay, so we can go ahead and add these tick marks for all of the characters that we've been looking at. In fact, we could add tick marks for any character that is in any of these organisms in this group, because we can reconstruct where along this phylogeny those traits evolved, assuming that we really trust this phylogeny. Once you've got those, you can reconstruct for these particular characters what any of these ancestors looked like. So for example, here's an ancestor you might be interested in. This is the ancestor of all amniotes. One of the really important evolutionary leaps forward among vertebrates was the evolution of what's called the amniotic egg. This gave animals, really for the first time, gave vertebrates for the first time the ability to reproduce away from water. It allowed these organisms, the reptiles and the mammals, to essentially colonize dry land and really take over in the niches where we see them today. Okay, but what did this ancestor look like? Let's suppose we don't have a fossil record of that ancestor. Can we reconstruct what its morphology was like? Well, yeah, up to a point. I mean, presumably that lineage that led to this point, started out looking like the outgroup, the lamprey, and then we just walk forward from the root to this node. So we're kind of going along here, it's evolving. Oh, okay, so we've got a lamprey, except it lost its fins. So, all right, we've got an organism without a fin. We're going to keep going here. Oh, it lost the gills too, and it got claws. So that's what we've got. It's not a lamprey anymore, but whatever the ancestor to all the amniotes was, it didn't have mammary glands or fur, it wasn't warm-blooded, Didn't go through metamorphosis or have keratin scales or gizzard, but it did have claws or nails. It didn't have fins, and it didn't have gills. Okay, this may seem kind of trivial, that we can reconstruct a few characters for whatever this ancestor was here. But when you get a lot of characters and a lot of species, you can do much more powerful things with this approach. So, for example, a study that came out a few years ago, what scientists did is they took the sequence for the rhodopsin gene in living reptiles, including birds. And they reconstructed the genetic sequence. Actually, I think it was probably the protein sequence, but it was either genetic or protein sequence for the rhodopsin gene for dinosaurs. Okay, so they knew what the sequence was for birds, they knew what it was for crocodiles, they knew what it was for lizards and snakes and tuataras, and they used all that to say, okay, this is the most probable sequence for this protein in dinosaurs. What was neat is that they then put together that protein, and they actually, I think what they did is they cloned that gene into a cell line and got that cell line to express this particular variant of the rhodopsin gene. And they took it into the lab and they exposed it to different wavelengths of light and saw what it reacted to. And what they found is that this rhodopsin molecule is most sensitive under low light conditions. The implication being that dinosaurs could see in the dark. Okay, so what scientists basically did is from the information in living day organisms, they reconstructed a gene that has not existed for millions of years. And from the behavior of the protein that's made by that gene, they were able to infer something important about the habit of organisms that have never been seen by any living human. As we get more and more genetic data, we're going to be able to do more and more of this. In fact, we're shortly going to be getting to the point where we will be able to reconstruct the entire genomes of organisms that have been extinct for a very long time. And what this could do is it could open up all kinds of exciting doors to our understanding of how the organisms that we see on Earth today have evolved.